Very good morning, students. So today we are taking up the next topic under muscular system, and this is the cardiac muscles. So first of all, we should know that cardiac muscles are involuntary in nature. So in the very first topic of the classification, I had explained what is involuntary. So these are not under our conscious control, so that's why they are involuntary. Secondly, they are striated. That means they have got dark and light bands. So third major character is that they are present only in the heart. So that is the location. So they are present only in the heart. They are not present in your legs or your arms. These are striated. They have got dark and light bands. So they are not under your will. So heart is pumping the blood without your will. You are not controlling your heart. And fourth one is that they have got intercalated discs. So that is again a peculiar feature. So intercalated discs are the discs which are present in the cardiac muscles. Now I will be later on telling what is the intercalated disc and what is the function. And another major important characteristic is that they are branched. So they are branched in structure. So they are joined to one another by the intercalated discs that contain the gap junctions and desmosomes. So that is the important thing to know. So if I draw, suppose a part of cardiac muscle, you can refer to the notes which I provided to you for cardiac muscles. So this is the intercalated disc which is present here. So this is joining this cell with this cell. So this is a nucleus of cell number 1. This is a nucleus of cell number 2. So this intercalated disc is joining first cell with the second cell. So if I take, suppose this is cell number 3, this is cell number 4, this is cell number 5. So these are the intercalated discs which are joining these cells. So these intercalated discs, they have got two things. Number one, they have got gap junctions and they have got desmosomes. So you all know that desmosomes are like helping in joining two cells together, and they also have gap junctions. Next thing is they are very similar to the skeletal muscles. Cardiac muscles also have got the same actin, myosin, tropomyosin and troponin. So all the proteins which are present in the skeletal muscles, they are present in cardiac muscles also. Moreover, they also have the sarcomeres as the skeletal muscles do. So that means cardiac muscles and striated muscles, they are very similar in structure. Only the difference is that intercalated discs are present, and secondly they have branching. So these are the two differences. Otherwise, all of the proteins present in the skeletal muscle, the sarcomeres, the main structural similarity is the same. Now, taking up the difference, the cardiac muscles, they are shorter than the skeletal muscle fibers in length, and they usually contain only one nucleus, which is located in the central region of the cell. So, as we have already discussed, if number 2 cell is this, then it is containing only one nucleus. Otherwise, in the skeletal muscle cell, you can have many nuclei. So the cardiac muscles, they also possess many mitochondria for like taking up more of the energy. So as they require more energy, they have got huge numbers of mitochondria. Now, cardiac muscles are basically of two types. First type is the conducting cardiac muscle cells. So these are the muscle cells which are only present, I think, less than 1% of the total cardiac cells. So these are of larger diameter, and they are not producing any kind of tension, or they are not producing any kind of force with which the heart contracts, but these are conducting in nature, and they are only specialized for the excitation of the cardiac muscles. So they constitute a network in the heart which is known as the conduction system. So we have already gone through with the topic of the conduction system. So we got SA node, AB node, bundle of His and Purkinje fibers. So these contain the conducting cardiac muscle cells. Secondly, the second type is contractile cardiac muscle cells. So these are the cells which are making up the tensile force for the pumping action. So what the conducting cells they do, they conduct the impulses to the contractile cardiac muscle cells, then they contract, and then the heart pumps the blood. So ultimately, the heart is composed of these two kinds of cardiac muscle cells. Now students, something about intercalated discs. So this is a very important question, and you can get a dedicated question on intercalated discs. So basically, cardiac muscles, as we know, they are branched. So they are connected to one another at their ends by these intercalated discs, as we have discussed earlier. So an intercalated disc allows the cardiac muscle cells to contract in a wave-like pattern so that the heart can work as a pump. So what intercalated discs they do is, like, if we have got two cardiac cells, this is the cell number 1 with a nucleus, this is the cell number 2 with a nucleus, and this is the intercalated disc in between. Now this disc is joining the cell number 1 with the 2, 2 with the 3, and 2 can be with the 4, as it is branched, so it can go downwards also, can go straight, can go on an angle also. So this intercalated disc has got two things. First was the gap junctions, second is desmosome. Now first thing should be clear that intercalated discs are joining cells with each other. Now what is the role of gap junctions? So gap junctions, they are forming channels in between the cells. So suppose if this is a gap junction, it is just like this. So if it is a gap junction, it is forming a channel which can conduct some of the cations, positively charged ions, from one cell to the other. So it is just like sending some impulses from cell number 1 to cell number 2. So if we delete all of the gap junctions from this site, cell number 1 cannot pass the impulses to cell number 2. So in this case, the heart muscles, they cannot contract simultaneously or synchronously. So that's why the gap junctions, they are very, very important. So this is in between cell number 1 and 2. So again we have got another intercalated disc, and again the ions will be passing from cell number 2 to 3, and that's how the whole of the system, the whole of the muscular cells, they got activated for contraction. Now next thing is desmosome. So these are the two things which are present in the intercalated discs. So desmosomes, these are the cell structures that anchor the ends of the cardiac muscle fibers. So desmosomes, if this is a desmosome, it is like fixing these two cells on this place. So the cells, they do not pull apart when they are under stress, or they are beating, or they are contracting. So desmosomes, they help to keep the cells in the position, and wherever the gap junctions, they are helping in the conduction system in between the cells. So that's why intercalated discs are very, very important in cardiac muscles. The next concept is the formation of syncytium. So when a number of cells, they are connected with, suppose, intercalated discs. So this is the intercalated disc 1, this is ID2, this is ID3. Now they can take up the action potentials from one cell or the neighboring cell. So all these cells, they will be contracting almost at the same time. So this group of cells which are contracting simultaneously with one impulse is called as the syncytium. So the network of electrically connected cardiac muscle cells, it creates a functional unit which is called as the syncytium. So you can get 1 marks question on syncytium. Next thing is the control of the heartbeat. How the beat of the heart is controlled. So as we all know that we cannot control the heartbeat, as all the muscles are involuntary in nature. So a particular phenomenon which is called as autorhythmicity acts on the heart. So some of a particular muscle cells which are present in the SA node, they are activating the heart, or they are making the impulses. So these are under the control of autonomic nervous system. So they can speed up the heart or they can slow down the heart. So some of the hormones which are present in the body, or they are secreted in the body, they also can affect the heartbeat, the behavior of the person, or the environment of the person also affects the heartbeat. So SA node is acting as the pacemaker in the heart, and it is making impulses at the rate of like 80 impulses per minute. So that's why our heart beats at this rate. Now the next concept in cardiac muscles is that they have long action potentials. So this is a very peculiar feature of cardiac muscles that is not present in the skeletal muscles. Now what are these long action potentials and why they are present only in cardiac muscles? Now we are discussing this part. So contraction in each cardiac muscle fiber is triggered by calcium ions, as in the skeletal muscle also. So these calcium ions, they are coming from two sources rather than one. So here in cardiac muscles they have what? Two sources. But in skeletal muscle we have got only one source, which is the sarcoplasmic reticulum. But here the two sources are, first will be the sarcoplasmic reticulum, as in the case of skeletal muscles. But here in cardiac muscles we got the second source. So these are the voltage gated channels. So these are the specific voltage gated channels which are for calcium ions, and these are present in sarcolemma, means the membrane of the cardiac muscle cells. So now let us make a graph. If this is the membrane potential on one side, and here we have got time in seconds. Now if I make a graph like this, that means here the action potential is formed. This is the increase in action potential. Now why? Because at point number 1, calcium channels, they are supplied to the cardiac muscle cells from sarcoplasmic reticulum, and after a very short period the calcium ions here, they are taken back into the SR, and the muscle action potential is lost and the muscles, they relax. So this is a very common kind of a graph which is present in skeletal muscles. Now we are taking up the long action potentials. Now what is the mechanism of long action potentials? So here, along with the calcium ions, sodium ions, they are very important in cardiac muscles. So here the graph is just like this. Axes are the same. So if I take up the graph, it is just like this. So why I'm saying long action potential is due to this part. So the muscles, they can remain contracted for longer times as compared to a skeletal muscle. So what happens here is that at this point, sodium channels, they are open. So what happens that if sodium channels are open, they can make up impulses. So they generate an impulse. So this is the membrane potential. So if sodium ions or sodium channels are open, they can generate a membrane potential. So on this point on the top, what happens that these sodium channels, they close. They are closed. But when they close, potassium channels, they open. Now here two things are happening. First are that sodium channels, they are closing, and potassium channels, they are opening. So the membrane potential is lost to some extent due to the closing of sodium channels, but it is not dropping as such down as this case. So this is because of the potassium ions, as these channels are now open. Now at this point, the significance of calcium ions is recorded. Now at this phase, the voltage gated calcium channels, they open, and more of the calcium influx, it occurs, and this results in a sustained membrane potential, which is also called as the plateau. So why we call them as long action potentials? Because they have got plateau. So all the muscle cells or the cardiac muscle cells, they are having this plateau due to the opening of voltage gated channels of calcium ions. So this membrane potential, it is due to calcium and potassium. Now at this point, what happens? The calcium channels, they close, but remember, potassium channels are still open. So that's why it is not lost just like this. Due to potassium channels, or due to the presence of potassium, this line goes like this at an angle, but at this point, all the channels are closed. So that's why the membrane potential comes to the same level from which it started. So if you knowing this particular graph, then you can discuss the long action potentials in the cardiac muscle cells. Now I'm trying to restudy this graph. At point 1, the sodium channels, they open, and sodium channels now they are present in the muscle cells. So we have a membrane potential. At this level, after some time, sodium channels, they close, and potassium channels, they open at this point. So there is a drop in the membrane potential. So after certain time of this gap, the voltage gated calcium channels they open. So now we have got potassium and calcium channels open at this point. So due to the presence of calcium and potassium ions, the membrane potential is decreasing but is not going this way. So there is some potential due to calcium and potassium ions. So it is decreasing, and after a gap period of this much, here the calcium channels, the voltage gated calcium channels, they close, and now we have only the potassium channels open. So the membrane potential decreases, and at the last point this will be 2, 3 and 4 and 5. So at number 5 we have all the channels closed. Sodium is also closed at 2, potassium is closing at 5, calcium is closing at 4. So the membrane potential decreases to 0. Not specifically 0, but to the resting potential. Now the next concept in cardiac muscles is the refractory period. It's again a very important part, and you may get a question on refractory period in cardiac muscles. So this is the time period during which the voltage gated sodium channels, they are inactive. So the time for which the sodium channels they are inactive, it is called as the refractory period. Why it is called as a refractory period? Because in this duration, if you give any stimulus or impulse, that will not cause any contraction in the cardiac muscles. So in this particular period, if you are giving any stimuli, like you give 2-3 stimuli to the cardiac muscles, they will not contract. So that's why this period is called as the refractory period. If I have to tell you with a graph, just make a graph which you have discussed earlier. So that was just like this. That was the point 1, this is the point 2, 3, 4 and 5. So we have the calcium channels, they are opening on this point, and calcium channels they are closing on this point. So that was the plateau period. So in this period, here on point 1, the sodium channels were open. Here at point 2, the sodium channels were closed. So just like from this period where the sodium channels are closing, at this time the refractory period starts. Now from this time to, if we take the time up till the calcium channels are closed, this period of time, it is about 180 milliseconds. Now this period is the refractory period. In this period, if you are getting any stimulus, it will not leading to the contraction of the cardiac muscle cells. So with this graph you can give the concept of 180 milliseconds of refractory period in cardiac muscle cells. If I take up the tension criteria in this graph, then from point number 1 we can draw, I'm drawing it with a black pen. This will be the graph for the tension produced in the cardiac muscle. Now if I relate this particular tension curve with this graph and with the refractory period, now just look at this graph. Here it simply means that when the sodium channels they open, there is a stimulus to the tension in the cardiac muscles. Now when the sodium channels they close, they are giving they are stimulating the calcium channels to open. So when the calcium channels they are opening, there is a brisk increase in the tension. So this curve is for the tension in the cardiac muscles. So now they are contracting. That means this is the curve for the contraction or the tension or the force which is produced in the cardiac muscles. This graph is not for the force. This is the membrane potential. Just see the difference. This is the impulse, or this is the membrane potential, and this is the curve for the tension. Now if I take one tension of a cardiac muscle, suppose this is one beat of the cardiac muscle or the one beat of the heart, it is corresponding to a particular wave of a membrane potential. So we are talking about the refractory period, and this is the refractory period. This is of 180 milliseconds, and now heart is contracting. It is under systolic contraction. It is under systolic contraction. This is the refractory period, and if I am giving one impulse in this period, another impulse in this period, third impulse in this period, do you think the heart will contract again and again and again? No. When this cycle, one heartbeat will be completed on this point, then you can give another stimulus. So that's why the heart is not under tetanus conditions, as in the case of skeletal muscles. In skeletal muscles, we can give many impulses before the relaxation of the muscle. But here in the heart, when this beat will close, then you can give the second impulse. Otherwise, in this period, you cannot give any of the impulses, and this period is called as refractory period. Now I think you should be clear about refractory period. So students, that was all about the cardiac muscles.